Good afternoon. My name is Will Valdar, and I'm going to be talking about modeling gene by treatment effects, gene by treatment interactions in the collaborative cross and other genetically diverse replicable populations. By gene by treatment interactions, I'm speaking very broadly here. So a gene would be a gene or genomic region or a genome. Treatment would be drug, maybe a therapeutic intervention, some kind of environmental insult like a viral infection or a diet. These are just examples. And G by T is where the effect of the treatment varies with genetics. And depending on the field that you're in, you might call the study of G by T pharmacogenomics, toxicogenomics, precision medicine, G by E, etc. And I'm going to define G by T discovery as QTL mapping to find G by T relevant genes. The list of treatments that I gave here are not random, rather these are ones that we've been working with in my lab, and we've increasingly been doing G by T studies. And so during this talk, I just wanted to share some thoughts and observations on doing these kinds of studies. First, I'm going to talk about some general G by T concepts and introduce the notion of delta. Then I'm going to briefly describe some G by T studies in the collaborative cross on drug-induced liver injury. And then I'm going to share some observations and thoughts about delta. So the first thing to point out is that G by T discovery is hard. It's extremely hard to do in humans. That's why you don't see many GWASs of gene by treatment effects. And I would argue that it's hard to do in outbreds more generally. And the reason why is that in the standard genetic analysis, you're associating genetic state with a directly measured phenotype. But in a G by T study, you're associating genetic state with treatment response. What is a treatment response? Well, treatment response means the causal effect of a treatment. And to think about this, we could imagine two individuals. One you assign the drug treatment to, and the other the placebo, and then we measure the outcome of interest, which will be Y1, in fact I'll call it Y1 drug and Y2 placebo. And at first glance, what one might think, well, the effect of the treatment is the difference between Y1 and Y2. But that's not quite the right way to think about it. In fact, what we need to imagine is the counterfactual. So what if we had given individual 1 the placebo and individual 2 the drug, and observed this counterfactual Y1 placebo and Y2 drug? So we're actually looking at 2 treatment effects. One is the causal effect of the drug on individual 1, which you get by subtracting Y1 placebo from Y1 drug, and then by similar process, the causal effect of the drug on individual 2. Obviously, you can't do this because it involves things that you can't see. And a fundamental problem of causal inference is that the causal effect of a treatment can never be observed. But we can estimate an average treatment effect, but only when 1 is a good match for 2, only when the 2 individuals act as kind of counterfactuals for each other. So a natural version of this in mice would be paired mice from a single strain. So we could take 2 same-sex litter mates, basically a similar as possible, and we could randomly assign the drug to one and the placebo to the other. And if we labeled them I and I prime, then our treatment response estimate for that pair would be delta equals yi drug and yi prime placebo. So we end up with this delta, and we could get a better estimate for what the strain delta is by doing more pairs. But the point is, the unit now is not the phenotype, but it's the delta from a pair of mice. If we did this in 2 strains, then we could calculate a gene by treatment effect by looking at the delta, the estimated delta for strain 1 versus the estimated delta for strain 2. And more generally, if we had many strains, then we could fit a sort of general regression model where the dependent variable is the delta for each pair, and then the intercept is just the overall treatment effect, and the strain is the G by T component. So in order to do something like this, you obviously need lots of strains and to span a lot of genetic diversity if you want to do G by T discovery. And you need strains because you need biological replicates, and you want genetic diversity in order to cast a wide net regarding the QTL mapping. And so a good population for this is the collaborative cross. This is a panel of recombinant inbred strains that are descended from a genetically diverse set of 8 inbred strains, and they encompass more genetic diversity than the human population. So the 3 drug studies that I'm going to talk about, one was on a drug called tolvaptan, one is a drug that's still in progress, and the third is idelalisib. And these studies took the form that I've just described. In fact, there were 4 pairs measured per strain, and there were 45 strains, 44 strains, 50 strains, and in 2 of those studies we measured microarray expression, and in all of the studies we looked at biomarkers of drug-induced liver injury. I'm not really going to say anything more about those studies in general. You can read more about them in these papers. The bottom line is we found some QTLs, we found some candidates. Interestingly, the power was lower than for a typical kind of phenotype mapping study. One of the parts of those studies that I found really interesting and that really, to me, brought home the difference between considering directly measured phenotypes versus deltas, was the eQTL mapping. And we didn't really talk about this in the papers, but first of all, let's, we did eQTL mapping on the placebo-treated only mice, and this is a very typical kind of eQTL mapping. It's just on the directly measured transcript levels, and you see a common picture here, which is the heat map of the eQTL mapping, with QTL location along the bottom, transcript location along the side, and then a diagonal line showing all of the cis QTLs. So that should be very familiar for people who've looked at these kinds of plots before. If we just looked at the drug-treated mice and did the same thing, we see a very similar picture. It's not exactly the same pattern, but we see the diagonal. And I should note that there are some drug injury studies that have just used drug-treated mice. Okay, but because of the way we designed our studies, we were able to look at the delta. And when we map on the delta, instead, we see a quite different picture. It's much more sparse. There's no cis-QTL line. And the first time we saw this, we thought, well, maybe that's some kind of mistake. But then we realized, oh yeah, that actually makes sense. My slide. Yeah, okay. So here are some observations that we get from that. When we study treated mice only, the treatment response is confounded with baseline. And so the results that we see may mostly reflect baseline effects, not the treatment response that we want to see. If we use delta values, that does tell us about the treatment response. And what we also see, though, is that genetic effects on the treatment response are few. They're detected with low power, which could be the reason why we see not very many of them, and they're estimated with less precision. Why is that? Well, if we think about a kind of model for the placebo phenotypes as being mean, genetics, and noise, and then a model for the drug phenotypes as mean, genetics, and noise, then the delta is really the combination of those two things, one thing minus the other. And importantly, the noise term is drug noise minus placebo noise. And if you're used to thinking about variances, then you'll know that when you take one type of noise and you subtract a different type of noise, you don't get less noise, you get more noise. The noises add together in their variance. And for that reason, working on the delta is noisier than either working on the drug or the placebo-treated mice. This means that the studies are going to be lower power, and this has implications for experimental design. A good thing is that delta avoids having to separately model the two different sorts of noise, or having to assume that those two sources of noise are just as noisy, as would be done in traditional interaction models. So in summary, G/T discovery benefits from a population that is replicable and genetically diverse. And some good options for this are the collaborative cross, but also other inbred strain panels. Rigorous G/T discovery requires both treatment and placebo. It's inherently more noisy and low powered, but this just means that you have to design in a way that mitigates that and model in a way that takes advantage of that. I'd like to thank these people, in particular my student Yang-Wei Kai, and my collaborator Mary Mosedale, and thank you for listening.